Okay, physiology of diabetes. We talked about the scope, the characteristics, and now I'm going to attempt to link obesity and diabetes, but mostly to give you a sense of what goes wrong in the muscle, in the body with diabetes. And it's probably going to be similar to the message you've heard before. Diabetes in its simplest form is dysregulation of glucose, and that's no surprise. Dysregulation or inability to properly handle glucose. And what I mean by handle is take up, dispose of, and store. We need to do that because free glucose is actually quite damaging to the epithelium, to peripheral nerves, and that's why neuropathy, ulcers, etc. are large consequences of severely progressed diabetes. They don't occur right away, but the accrual of damage from persistent high glucose will result in an increase in the incidence of neuropathy and ulcers. Epithelial tissue seems to be really susceptible, so we don't want to have too much circulating glucose, free glucose in the blood. We want to get rid of it. And our body has a great mechanism for doing just that. You know that as the insulin response. So if we take in a meal, specifically with carbohydrate, that triggers the release of insulin from the pancreas, and insulin is the signal to take up, dispose of, and store that glucose. Now, dispose could also mean oxidize and use for energy. That's not the signal that insulin confers. Insulin is a rest and digest signal. Store it as glycogen in the liver and the muscle for future use. And so normally, insulin's released, the muscle reacts, we dispose of and store most of the glucose in muscle, some in the liver. And these characteristic traces, the red one on the bottom, shows you what happens in response to a meal. We see a spike in blood glucose that's quickly brought back down to normal, which is this horizontal solid line. And glucose is probably one of the most tightly regulated variables in the body. We don't want it to stray too far from this normal line. That's why the quick response. But we're realizing in recent years we can forcibly manipulate this response by continually insulting the body with a high glucose, high insulin, high energy type diet. We get this resistant or compromised response, shown here by the top line, where in a diabetic individual we, 1, start higher, 2, we see a larger increase, and 3, that increase persists much longer than in a normal healthy individual, and that's increase in blood glucose concentration. So overall, the response is compromised. And these are on the same figure, they're on the same scale. Higher starting point, larger inflection, larger increase, and more prolonged, sustained response. So something about the signal that insulin conveys is compromised. And the question that we want to answer in this chapter is what causes us to move from this red line to the green line as we become insulin resistant and diabetic. So there's a suboptimal response. And I suppose I'm of the opinion that this suboptimal response is due to the excess storage of energy, the development of obesity, and possibly any number of other characteristics. Obesity is related to a whole host of factors, and you've seen slides like this before where there are linkages between obesity and diabetes, hypertension, arthritis, depression, cancer, and it's not to say that it causes all of the items on this list, just that they tend to appear together. Perhaps obesity causes diabetes, and that's what I'm going to try to argue to you today. Perhaps it causes sleep apnea like we saw in the COPD section. Not sure if it causes cancer, but they are often associated or present together. There's still a lot of research in that area. It could be a side effect of cancer, for instance. But as far as obesity and diabetes go, the two theories that we are going to discuss are one, that of what we call quote-unquote lipotoxicity, toxic reactive lipids. There's some accumulation in the body, some accumulation in the cell that disrupts normal function. And disrupts, we'll take lightly for now, somehow it disrupts normal function. The second theory might not be separate from, might be in addition to the first theory. That somehow could be because of mitochondrial dysfunction, a backlog of the processing of energy in the mitochondria, and then negative signals associated with that backlog interrupt function in the cell, upstream in the cell, perhaps by the insulin receptors, who knows. But the signal originates down in the mitochondria because of some forcing of energy or a backlog of electrons trying to be forced through that system. And we'll look at that in detail. So two main theories, and there's probably some new evolutions to these theories. It's actually been a while since I've been in this area, a couple years. But the information that we talk about was agreed upon, maybe not conclusive, but certainly agreed upon at the time. Yeah, Leanne.
So this is theories to resistance, or why obesity causes?
These are the theories to how diabetes develops, and we're linking that with the progression of obesity. So the accrual of fat in reactive lipids, or the accrual of fat in creating a backlog at the mitochondria, are reasons why obesity can link to diabetes. And it's through these two mechanisms. These are the two linkages, if that helps. So let's diagram lipotoxicity first. And this is a simplified drawing of the insulin cascade. Highly simplified, even though it still looks fairly complicated. There's some acronyms here that we're not too sure of, and you don't need to know insulin receptor substrate and PI3 kinase, AS160. These are steps in the insulin signaling cascade. It's been well characterized. And in a normal, healthy individual, it plays out as follows. Insulin binds the receptor, causes phosphorylation of the receptor, which initiates a cascade of phosphorylation to turn on the downstream enzymes. Ultimately, somehow the signal is sent for glucose transporters that are stored in the cell to move to the membrane, and then when they are at the membrane in contact with that circulating glucose, they will take it up to be stored within the cell. And this, for our purposes, could be a muscle cell. Insulin binds to the receptor, the signal gets sent down. GLUT4, which is our candidate glucose transporter in muscle, actually moves, which allows it to do its job of taking up glucose. This is a normal situation. Lipotoxicity relates to the fact that as you become insulin resistant and or diabetic, you also tend to increase consumption of total energy and specifically consumption of fats. So here we have free fatty acids also circulating with glucose in a meal that you might have eaten. Free fatty acids are also taken up by the muscle cell through a transporter, and we're not going to go into depth about what that transporter is just yet. They need to be processed and stored just like glucose. They can be oxidized, they can be stored for later. They need to be tagged or activated. This coenzyme A is a way that in the cell we tag the free fatty acid for a certain role. And often that role is to be stored as triglycerides. Triglycerides are an inert, stable form of fat storage. We want this to happen. We have no problems if we we take up fat and store it as triglycerides. What we have a problem with is if fat is taken up and stored in other forms. So triglycerides, 3 free fatty acid molecules attached to a glycerol backbone. Normal, inert, stable storage, that's good. Diacylglycerides, on the other hand, not so stable. Only 2 free fatty acids bound to that glycerol backbone. There's an open spot. The shape is different. The function is different. Diacylglycerides are an intermediate. They're a step towards triglycerides, but they have their own activity in the cell. Ceramides are a processed form of that fat that doesn't get bound to glycerol, that's not stored as triglyceride. The free fatty acid is converted to a molecule that we'll call a ceramide, and these two things are less desirable. They're less desirable. We want them to be stored as triglycerides because it's inert and stable. If they're not, there's disruption that can potentially occur. And there's a series of studies that looks at if and how the appearance of these two enzymes, or not enzymes, these two products specifically affect insulin function. And there's a good idea that if we see diglycerides in the cell, they disrupt, even upstream of that, just free fatty acids in the cell disrupt phosphorylation of the insulin receptor and the proximal stages of the insulin cascade. Diacylglycerides also disrupt the early stages of the insulin cascade. Ceramides disrupt downstream the later stages of the insulin cascade. And the disruption is generally proportional to their abundance. More of them, more disruption. That makes sense. The consequence of this disruption, if fewer enzymes are activated, less cascade occurs, less signal gets through, fewer glucose transporters move to the membrane. It has nothing to do with the appearance of insulin or the number of receptors. It's transmission of that signal after being received. The same signal has less downstream effect. This is insulin resistance, right? In its pure and absolute form. And this, as I diagram it, is what we consider lipotoxicity, the fact that these intermediates can somehow disrupt function. And maybe it's because they are shaped in a similar way to the enzyme that wants to activate this cascade. Or maybe they're shaped in a way that it knocks the phosphate group off and inactivates these enzymes. We're not sure, but somehow they disrupt signaling. I think it's important to note that while I'm demonizing diglycerides and ceramides on this slide, if you have a supply of fat in the diet that is taken up by the cell, and one of these increases, they all tend to increase in parallel, right? So if we have one, the abundance of all of them will increase. So if we increase the intake of fat in the diet, all of these forms of fat will increase in the cell. And that makes the most sense when you consider diglycerides, because this is the step towards triglycerides. The fat comes in, binds, makes a monoglyceride, adds another fat, makes a diglyceride, adds a third free fatty acid, makes a triglyceride. And it's an incremental process, so this has to be a stepping stone on the way to storage. But if we have a surplus, an abundance of supply, then we'll have much more of all of these individual subspecies in the cell, and more of this disruptive effect in the cell. So do we see characteristics? Do individuals exhibit characteristics that would support this schematic? That's the next question. We have a nice diagram here that hopefully makes sense. Do we observe this in general? Well, if we compare individuals across a varying range of levels of insulin resistance, levels of obesity, we see a progressive increase in fat uptake. And I purposefully removed the label on the left-hand side. The numbers, you can take it for what they will. A higher number means a higher uptake. I'll describe briefly the process that we use to make these measurements. Uptake is notoriously hard to do. How do you ever measure something being transported across a membrane? You can't measure the activity of the enzyme. It's hard to get into the cell afterwards. So this preparation is really unique. This was done actually in a lab down the hall from me when I was doing my doctorate. So I got to be privy to a lot of how this information was created. You take a series of cells and then some chemicals to prepare them. And if you have a normal cell like this, a specific chemical will make it elongate a little bit, and then a portion will bud off. So I'll have this elongation, and then a little bubble of cell will pop off, and the rest will remain intact. And you do that enough, you have all these little bubbles called vesicles. And those bubbles are representative samples of the membrane. You have the entire cell, entire membrane, and then as a little bit bunches off, it should have all the proteins that would be in the membrane. So if you take these vesicles now and put them in a fat-rich or a carbohydrate-rich environment, then you wash the environment away, you break the cells up, anything that was in the cells is released, and you can measure what uptake would have been. So that's how we measure uptake in this scenario. We have muscle from lean, overweight, obese, and diabetic individuals, and those small little buds of the cell, those vesicles, will take up fat. They still work if they're provided fat, if the transporters are there. And all this says is that normal muscle in these conditions takes up more fat if you're diabetic, less fat if you're lean. And there's a general progression across the spectrum. So greater fat uptake in those obese and diabetic individuals. So we have the stage set for lipotoxicity to occur. Greater uptake of fat. If we believe that all of those subspecies will accrue together, it's difficult to measure ceramides or diglycerides. So in the lab that not that I was in, but that was in our department, they measured triglyceride content and used that as a surrogate for all of the subspecies. And with an increased uptake, we also observed a greater accumulation of fat within the muscle cell. This is now taking the entire muscle, chopping it up, and then measuring how much triglyceride was there. So we assume that this also means an increase in ceramides and diglycerides. But again, it supports the idea on the other slide. Now, this is really the operative finding, that there is a higher content of fat. And so we're kind of curious as to why this accumulates. Uptake is one half of the balance equation. The other half would be disposal, right? Or oxidation. Why is there an accumulation of fat in obese or type 2 diabetic muscle? It seems that in addition to having an increased uptake, these individuals have a reduced oxidation or a reduced disposal. So I always think of it as a sink. Increased uptake, the faucet's on full blast. Decreased disposal, you've got the plug in the drain or something's clogging the drain and the sink fills up. If you can enhance disposal, if you take the plug out of the drain, maybe the sink empties. increasing oxidation can help get rid of the stored fat. But that analogy, I think, holds up. They have less oxidation overall and a higher fat uptake. But what's really interesting is that if you don't use the muscle as a whole and you pick out the individual mitochondria, they still work pretty well. Mitochondria still work well enough. They can do their job of oxidizing fat, oxidizing carbohydrate, making ATP. It's just that it seems that obese and diabetic individuals have less of them. And maybe that's a consequence of lipotoxicity. Maybe it's a consequence of a sedentary lifestyle and a detraining type effect. But the decrease in oxidation seems to come from a smaller volume of mitochondria. They still work well, but in the muscle as a whole, oxidation is reduced. So it seems that the schematic for fat accrual in the cell goes as follows. We have a normal healthy cell where when fat is in the circulation, it will be taken up by receptors, but there's since been research saying there's actually vesicles inside the cell that contain transporters for fat. Just like GLUT4 moves to the membrane, fat transporters move to the membrane in response to a stimulus too. So if you consume a high-fat meal, these will move to allow uptake of the increased surplus of fat. Normally, we have some storage, we have some oxidation, and balance, a normal amount of triglycerides. But in an obese or diabetic individual, we see higher uptake and reduced oxidation. Higher uptake largely from a persistent, constant relocation of these transporters. They don't stay inside the cell, they live on the membranes. They're always available to take up fat, hence the increased uptake. And while on an individual basis mitochondria seem to work well, there's less of them in the cell. A reduced volume which plugs the drain and leads to the accumulation of these fat species that causes downstream effects, downstream inhibition of insulin signaling. So we're presenting this more as a fat problem where the consequence is dysregulation of glucose disposal. A fat problem where carbohydrates are the symptom. But this isn't that satisfying. To me, anyways. I believe that fat uptake is greater, and that we observe an accumulation of triglycerides for sure, and that probably also means diglycerides and ceramides. There's actually a good body of work that correlates all of these, and I'm not showing it to you here because it's just fairly complicated. All of these do accumulate in obese or diabetic muscle. But where I'm left wondering, where I'm not satisfied, is in how an increase in these stored fat species affects insulin signaling. Somehow it interferes with the cascade. The question is, how does the transition occur? I don't know that I worded that right. The question for me is, how does this signal cause a disruption? Somehow is not good enough. How does the signal from an increased prevalence of fat cause a disruption? It could be structure and function, it could be shape, it could be that the enzymes are trapped in a globule of fat. We're not sure, we haven't gotten that far yet. But the second theory of diabetes might provide a linkage. The second theory might provide a linkage. And that second theory, before I get into the second theory, does lipotoxicity make sense? Fat builds up, somehow gums up the works. Okay. Yeah, question?
In that, for mitochondria, you said there's a reduced volume?
Yes.
Is it a reduced volume for the increased workload, or does the actual number of mitochondria actually reduce?
So is the volume of mitochondria reduced in that it's lower than it should be? Or is the number reduced? Is that what you mean?
So let's
For
say
the activity level?
Yeah, for the activity level. Let's say that you would only have like 1 mole come through, so you have like 10
Okay.
mitochondria. Now do you have 2 moles for 10 mitochondria, or do you have 1 mole for like 1 mitochondria?
Okay, so I would say that when you isolate the mitochondria, the oxidation rates are similar in diabetic and healthy individuals. So I don't think that it's less efficient. I think they still work as well. But it's the number and the size that are reduced. And it's the same thing that happens with detraining. So my personal journey from last summer to this summer, a nice journey detraining. Mitochondrial volume decreases, fewer mitochondria, because you don't have the persistent high energy demand. You don't need to keep those mitochondria around to make ATP. And volume, because we're not sure if, actually we're the classic picture of mitochondria are these small olives in the cell, small little bubbles that make ATP. But we are leaning now towards them being connected in a network, sort of like the sarcoplasmic reticulum. So it's not technically correct to say the number goes down, because if they're all connected, it's one large reticulum. So the volume is a way to approximate. If it's a number, volume goes down. If it's the size of the reticulum, that also goes down. So volume just means there's less machinery to make ATP, put it that way. Yeah.
Like, for the transition to obesity, like, because when people are foragers and stuff, like, this would work really well, because if you happen to have a period of time where your only intake was fat, your body would be really good at storing it.
Yes.
Right? But then, and then it would interfere with the insulin signals, I guess, because people would be, like, very active, so, oh no, that would still be an issue.
It would still be an issue. I love your first point. That's my take on the issue as well. The blueprints that we have here serve us incredibly well if there's ever a famine situation or we go without energy. We want to store as much as we can. The schematic would work. You would become insulin resistant if you were a forager and you consumed a large amount of fat and a large amount of energy. But foragers typically didn't see that much fat or energy to be consumed. The search for food was a very active pursuit. There wasn't a surplus, an abundance of food. And so I don't think that they ever reached the point where they became insulin resistant, or if they did, it was temporary. If they gorged themselves on a mammoth one day and they took the next day off sleeping in a meat coma, it was transient, it was temporary, and they went back to being active and insulin sensitive.
Then, like, for modern day, the transition would just be that, like, our bodies are still really good at storing fat, but now we're not burning it.
Modern day, we haven't had time for our genetic blueprint to change. Our DNA is based on hundreds of thousands of years of evolution. In the past 50 or 60 years of abundance, we haven't been able to change that we like to store energy. Our body doesn't believe us. 50 or 60 years in the terms of glacial ice ages is a drop in the bucket. So it's not going to change the blueprint because it's still expecting the next famine to occur. That's my interpretation. I can't say with any certainty that actually happened. I wasn't there, but it makes sense. Yeah. Okay. So the second theory, I think, helps to create a signal. And that signal is loosely what I would call reactive oxygen species. And this is a class of signals from the mitochondria that are typically released in response to stress. Reactive oxygen species is even too specific. Free radicals might be a better term. Any molecule or substance or protein that has forced upon it too many electrons becomes unstable or becomes reactive. Oxygen is one of those. If oxygen has too many electrons, it becomes a reactive oxygen species or a free radical. and they erratically move around the cell and can bump into and donate those electrons to things that shouldn't have them, which alters their function, so on and so forth. Anyways, a signal in response to stress, which is actually a good signal. We always talk about ROS or free radicals as being bad, but you need this stress response for adaptation. And I love to bring up a whole body applied study looking at the training response, so normal exercise training, with individuals taking a heavy vitamin A, vitamin E dose of antioxidants versus a placebo pill, and the training response is actually blunted in the antioxidant group. They become less fit. It takes longer to become as fit. And we expect that that's the antioxidants disrupting this free radical signal. So we need this signal to be able to adapt to exercise. And while if it persists, it's negative, it's a bad thing, and it causes some detriment to the cell, the short-term stress that causes an adaptation to do better in future is actually good. And that short-term stress is what we call hormesis on the slide. Transient stress that prompts an adaptation so that the stress is lessened in future. So it could be, rather than fat somehow disrupts the enzyme activity or the signaling cascade, it could be that if the release of ROS, or the release of free radicals, is disrupted with obesity, and it's progressively more and more disrupted as you become more obese or diabetic, then these might be the conveyors. They might be the signals that link obesity and diabetes or glucose dysfunction. So we have to see if you would ever generate more or less ROS in response to a surplus of energy or fat in an obese or diabetic situation. Now, the way I imagine this happening, or the diagram that I'm showing you to summarize how this happens without showing you a bunch of complicated acronyms and figures, is as follows. So this is an enzyme that breaks down carbohydrate, or that signals part of the insulin cascade. It's a general enzyme in the cell, and enzymes often are turned up or turned down. They're turned on or turned off, and we do that through a series of signals. So the most common is phosphorylation of an enzyme. Phosphorylation is almost the go-to signal to turn an enzyme on, and dephosphorylation turns an enzyme off. There are exceptions where the opposite is true. You can dephosphorylate to activate. The point is, we're changing the situation to change the activity of the enzyme. And in the simplest form, we might phosphorylate one protein, one amino acid on this enzyme protein. We can also phosphorylate two locations. We could phosphorylate two locations and dephosphorylate a third location. Phosphate isn't the only signal. We can also methylate, add a methyl group. We can remove a methyl group. We can acetylate. And these are not the only activators or deactivators. So the picture that I'm trying to paint for you is that it's the net sum of these influences that dictate the activity of the enzyme. And really all this is doing, as we add a phosphate or add a methyl or remove an acetyl group, all we're doing is changing the quaternary structure of that protein, right? We're just shaping it slightly differently. We're opening up the active site. We're closing off the active site. We're letting glucose bind to it. That's all we're doing. We're changing the shape slightly. And you've probably heard that structure dictates function. If you haven't, that's a pretty universal truth. And so the sum of these signals is set up in a way that under the appropriate conditions, enzyme X is so active. When we have an abundance of glucose, the enzymes in the signaling cascade are turned on. If free radicals come into play, what happens is they donate their extra electrons to an enzyme, to a protein, and disrupt normal function. So disrupting normal function is now we don't have a phosphate or a methyl group that we would normally have. So normal situation, and then if free radicals are in the mix, that normal situation is affected like so. Boom. Okay? The signals are gone. The effect of removing those modifiers is then to change the shape of the enzyme. It's still the same enzyme. It still tries to do the same job. It just doesn't do it as well. It's shaped differently now. So now I have this differently shaped enzyme that's still trying to look for the same substrates and products and interact with the same proteins in the cell, but it just can't do it quite as well because it's shaped differently. And this is how free radicals or ROS generation would impact normal metabolism in the cell. So the question becomes, where do these come from? Where do ROS come from? How are they produced? And does their production change with obesity? And this is where I get to present these fantastic slides from Dan's old advisor, which are now quite a bit dated, but I think the message still rings true. You don't have these on Moodle. You don't have a copy of them in your slide set because he did. I expect he copyrighted them, but this helps us understand the production of free radicals from a mitochondrial energetics perspective. And so this is the electron transport chain. And you should know that the electron transport chain transports electrons by default as we input protons or energy into the system. And for electrons to be pumped out, flow has to occur. For flow to occur, for flow of electrons through the chain to occur, protons have to be pumped out. Those two things go hand in hand. And they're important in understanding the production of free radicals. So the pumping of protons out across the membrane is linked to the cascading, the movement of electrons through the chain. And we get the electrons and the protons to be pumped from food. All food that you take in is eventually broken down to reducing equivalents. NADH, FADH2. Remember those from biochemistry? We don't care about the chemistry here. We just care that that's the building block. Food gets broken down into NADH and FADH2. And then these lend protons and electrons to the electron transport chain. There's an input of energy here. Electrons move through the chain, protons are pumped out. And this cascading series of events occurs in real time. We have more electron movement. As we get to complex 3 here, we have another handing off of the electrons, which is coupled to pumping of protons. In complex 4, the same thing. And then we latch this whole mechanism by reducing oxygen to water. And this is a non-reversible reaction in physiologic conditions, so this can't run backwards. So this is normal function. And normal function would mean at some point, if no other factors are in play, we will reach a point where we've pumped out a saturating amount of protons. And here, it's not saturating yet, but there's an accumulation of protons outside. And just from simple diffusion, we know that it's a lot harder for these protons to move out against the concentration gradient, right? It's hard to do this work. In theory, you could reach a point where there would be too high a concentration outside that no protons would be pumped through and the electron transport chain would stop, in theory. Right? Because the pumping of protons is linked to the transport of electrons. If one stops, the other stops. Well, if this were ever to occur, there's no room for protons to be pumped, there's no room for electrons to move, and these proteins get saturated with electrons. There's nowhere for them to go. And so in theory, this is the situation that would allow the production of reactive oxygen species. We have an abundance of oxygen down here that's always being refreshed from the environment. If we have this saturated or reactive situation with a lot of electrons on the proteins of the electron transport chain, some can slip off. Instead of O2, we have O2 negative. This has more electrons than it needs. This is now what we call superoxide, a reactive oxygen species. This will run about the cell, wreaking havoc, transferring electrons to other things that don't need them, disrupting phosphorylation and methylation, disrupting insulin signaling cascades. So this is theoretical. This never happens in practice, right? The electron transport chain never stops. It might slow significantly, but there's always some leak. There's always some leak. So it never completely stops. It just gets very, very slow. What we're showing here, we've added a little diagram on the bottom right. This is how quickly are electrons moving through the electron transport chain, and what is the membrane potential. So if there's low flow, we have a very large membrane potential. If there's high flow, then the pressure is released. We have a lower membrane potential, the difference in charge between the two membranes. So this is representative of a sedentary state. Somewhat saturated electron transport chain, some proton leakage, very low flow through the electron transport chain. It might look something like this. Now we also always need ATP to satisfy sodium-potassium pumping, vascular changes, etc. So there's always some energy being used, there's always some movement through the chain, but we have a pretty high membrane potential here. Slightly lower than the theoretical maximum, but still fairly high. Imagine what would happen if you try to now force more NADH into this system. This is a pressurized system. There's very little flow. There's a large opposition to flow. There's a high proton gradient on the outside. The analogy of trying to force in more NADH is the analogy of overeating, forcing fat, forcing energy into the cell itself. So not only do you have slow flow through the electron transport chain, but you are making it hyper-reactive. You're forcing more electrons onto these proteins. You're increasing the likelihood that free radicals will form because there's still this opposition to flow from the high proton gradient. So this is how we think obesity leads to the formation of free radicals. The free radicals will disrupt signaling in the cell. Signaling is that of the insulin cascade, decreasing glucose uptake. So how do we fix this situation? Any thoughts? How do you fix this slow, sluggish, reactive electron transport chain?
You can get rid of some of those excess electrons.
Okay, how do we get rid of the electrons or the protons?
Protons, sorry.
Well, I guess the point is, your point is that you can do either. They're both linked, right? You can't divorce the two. So you get rid of either. You move the electrons through faster, or you dissipate the gradient, which is done how?
By creating ATP and, sorry, is there a phosphorylation?
Yes. Creating ATP, creating a need for ATP, and dissipating the gradient through ATP synthase apart from the electron transport chain. So we can harvest this gradient by increasing activity level, using ATP, which creates the demand to make new ATP. Now we have much higher flow. We have a lower membrane potential. This is a much less reactive potential that's less likely to create reactive oxygen species. This also helps to alleviate the pressure of forcing energy in to the electron transport chain. Overall, decrease the push. Oh yeah, the second aspect was decrease the push. Don't force so much energy into the electron transport chain through reducing equivalence. So we can make this situation less reactive by decreasing the push of energy in. The push of energy in is one that will try to saturate with electrons, encourage ROS formation. So if we don't push energy in, we don't get this ROS production. And we can increase the pull by increasing the demand or the need for ATP. That increases the flow of electrons, decreases the potential, and the likelihood that we'll produce reactive oxygen species here. And yeah, go ahead.
It's the type of energy that's pushed in. All right, does the source of the energy make a difference?
Good question. Very good question. Does the source of the energy make a difference? And the answer is no. What we're missing here is complex 2 because it's not a transmembrane protein that accepts FADH2. Both carbohydrates and fat make both NADH and FADH2. Fat simply makes more FADH2 than carbohydrates do. So both fat and carbohydrates will force energy in here on complex 1 or in complex 2 that we don't have on the slide. But both of them will force that energy in. Yes,
And
Leanne.
so that's why there's like a sugar epidemic where people used to say like don't eat fat, so everyone switched to sugar.
Possibly, but so I don't think that shifting the blame is necessarily the right way to frame it. Yes, there is a shift now away from demonizing fats to demonizing sugar, but I think what we're missing is we should demonize excess energy. Right? It's not just sugar or carbohydrates, it's not just fat. It's the combination of both and the excess energy that we're trying to load into the electron transport chain. And that's why any successful diet is not only eat raw foods, be ketogenic, have low carbs. It necessarily requires you to reduce your total caloric intake. I've never seen a successful diet that has not done that. Maybe you feel more full so you eat less voluntarily. Maybe you're on a strict regimented diet where you're counting calories. Whatever the source of the food, the total caloric cost goes down as well. And that's what I think the driving force is for losing weight and improving insulin sensitivity. That's what I think the major influence is. I'm sure that there's certainly a reason to have healthy, nutrient-dense foods versus simple carbohydrates and Twinkies. But I could show you a pretty striking case study that sort of flips that on its ear as well. Yeah, Odessa.
Would muscle acidosis increase the production of reactive oxygen species if there's more protons present?
Muscle acidosis, would that increase the production of free radicals? That's a good question, and it depends on, I think, where the acidosis is sensed. So all of these protons here are on the surface of the membrane. They don't typically diffuse out into the cell. And likewise, the acidosis from really fast metabolism, I don't think diffuses into the mitochondria. So I don't think those two are related. And so I guess the answer would be no in that case. That's a good question, though. Yeah. Yep.
If you were to just like tip the scales, so like if you were to still increase, like push energy into the mitochondria, but instead you were to increase the ATP demand.
Okay. So
Yeah.
okay, so if you didn't do both,
Yeah.
if you only did one and not the other, then the simplest way I can describe it is with this sliding scale knob over here. This would be higher,
Okay.
right? You'd still have the push of electrons in, even though you're relieving pressure. There would still be a higher abundance of electrons than there would be if you reduced the push in and increased the flow of electrons. Does that make sense?
Yeah.
Okay.
So kind of like diet's more important than exercise in this sense. That's kind of like the
Energy balances. Negative energy balance is more important than positive energy balance, or less
Okay,
negative
yeah.
energy balance. I'm not going to weigh in on diet versus exercise. I think they are both equally useful. Actually, we saw a good example when we talked about those studies. 20% energy deficit from exercise or diet, both equally effective in changing weight and insulin sensitivity over a year. It's the lack of energy. And then both together had a greater effect, but either or can work. Yeah.
Was there
Razan.
a case when the increased activity was too much, so much so that the protons were like emptying faster than it was actually, like it was
Wow.
transferred in?
Good question. Is there ever a case where you can completely dissipate the gradient? I have no idea. I would expect no. But I don't know for sure. I don't know if we have a sensitive enough measurement technique to be able to answer that question. But I would imagine the electron transport chain is very responsive to the appearance of 1, ADP, and 2, calcium. Calcium activates these proteins as well to turn them on and turn on the pumping of protons and the movement of electrons. I don't think there's ever a situation where they wouldn't work, which means I don't think there's ever a situation where the gradient would completely dissolve. But I don't know for sure. I can't say with absolute certainty.
Follow-up question.
Sure.
Like, type of exercise influence how then like anaerobic versus aerobic with that gene.
Okay.
How much protons is being pumped in?
Would the type of exercise affect the flow of protons through ATP synthase, I think is what you're asking. The analogy I'll use is what we've just talked about, increasing the total cost of exercise. So maybe on a minute per minute basis, flow would be faster with HIIT training, or perhaps even with really intense strength training. But then those might slow when the demand is gone. And so I'm going to think of it in terms of the total ATP requirement, which is the total cost of exercise. The area under the curve. The more area under the curve, the more the gradient was dissipated, and the more energy we had to use to keep electrons flowing. So on an instant-by-instant basis, I'm sure there are fluctuations where one's better than the other, but I think overall, more energy use is better than less. Man?
But to completely dissipate the gradient, wouldn't that be like in a case of starvation where you like your body's breaking down all your tissues to use for energy, and then you have none, and then you die?
In an extreme sense, that would happen if you ran out of energy. But there's so much energy stored in your body that I don't know if you would ever die from using the energy you have on hand before you die from, what else can you die from? Oxygen deficiency to the brain, or cardiovascular complications with the heart not pumping. I think that you would still have energy on hand afterwards, and maybe a not a perverse, but a bad example of this would be in cremation. There's still energy left in the body because there's combustion that occurs when you cremate a dead body. I don't think you ever run out of energy available to satisfy these requirements. And even if you don't have enough glycogen in the muscle, you'll use amino acids to make substitutes. You can use fat to make rough substitutes, or you can still force NADH and FADH2 in from fat. I don't think you run out of energy. But the theoretical aspect is well taken. Yeah. Yeah, Razan?
Would lactic acid influence the amount of total energy that you can do because it hinders how much you can push yourself at one point? So would that
Yes, so this is less of a mechanics question and more of the applied physiology. The acidosis with high intensity exercise will shut off contraction apart from influencing the mitochondria. So there are signals that stop you from exercising that are not necessarily related to this, but it would decrease the total energy you would use because you wouldn't be exercising. You would have fatigued and you would stop. Yeah. Great discussion. All right, so I don't think we're going to see the video that I wanted to show you today, but we'll talk about some of the research in the alternative therapy section. We still have some time. We've already done the solution to obesity. 1, minimize the push of new energy in, so decrease the abundance of those reducing equivalents. Decrease the push in, which is essentially eat less. Lowering intake would reduce the potential storage of energy, or the potential push of energy into the mitochondria. The potential to make reactive oxygen species. 2, create more pull of energy through the electron transport chain. Use stored energy. Dissipate the gradient. And 3, do this persistently. You won't run out of excess stored fat with one bout of high energy demand or one bout of low energy input of dieting. Do this persistently. Create a persistent negative energy balance which would eventually drain the sink, which would eventually get rid of stored energy on hand. The bonus is we also get rid of any of those disruptive fats that are on hand, so we satisfy lipotoxicity as well by creating this persistent negative energy balance. We increase or decrease the likelihood of ROS formation, thereby decreasing the influence on insulin signaling and restoring insulin sensitivity. Eat less, do more is what this boils down to. And oftentimes I think, am I concerned that all of my training and my degree really boils down to eat less, do more? No, it just helps us relay that information to the general public perhaps. But we understand there's a lot more behind the scenes. We understand what it means to eat less, do more. And here's an example perhaps of doing more. And this is sort of a remnant from the previous iteration of these slides when we did a lot of prescription and the primary research discussion. So this is a typical cardiovascular prescription program. Exercising daily, meeting your MET minutes per week, satisfying a caloric goal, starting light and then gradually ramping up, light intensity walking, eventually moving to moderate intensity, short durations, moving to long durations, while also, to Dylan's point, considering dietary energy restriction, not simply eating all of the cheeseburgers and pizza that you want while exercising, although if you're not in a crisis situation, maybe you can leverage a few of those days, like I do myself. So now I want to take a look at alternative cures, because the solution to obesity as presented here is, it's satisfying in that it's effective, but it's discouraging because it doesn't happen immediately. And that is the problem for a lot of this knowledge translation to the general public. The therapy or the cure of eat less, do more is a long, drawn-out, habitual change. But there are alternatives, however extreme, that do show quote-unquote cures, or might be quote-unquote cures for diabetes. And so the first example of which is to take eat less, do more to the extreme. Excuse me. So reversal of type 2 diabetes in obese individuals with a focus on insulin production and triglycerides over 8 weeks, where these subjects ate only 600 kcals per day. So eat less, do more, really just eat less, but eat a lot less. And over 8 weeks, massive reduction in body weight from over 100 kgs to 88 kgs, 15 kilograms lost per week. That's almost 2 kilograms per week. 4 pounds per week, which is a lot. Normal glycemia restored in 1 week of this very low energy diet. So we saw in these individuals they exhibited typical high resting insulin, high resting glucose. They would have also exhibited that delayed resistant response to a glucose test. Within 1 week, normal fasting glucose levels. Paralleled by an equally impressive reduction, although not as abrupt, in here liver fat content. And the same occurs for pancreatic fat content. And they didn't measure muscle fat content. But if we use this as a surrogate for just fat content of the cell, progressive decrease where you'd say at 4 weeks it's pretty much normalized. And this could be a surrogate for energy content of the cell. This has inroads to explaining perhaps there's a benefit here on the lipotoxicity side of things, less disruptive lipid species, much improved glucose handling, reduced body weight with a very low energy diet. 17% reduction in body mass. Nuts. This is extreme, but not the most extreme. This was a case study. Oh, I have to refamiliarize myself. How long was this? 382 days, over a year, not eating. 0 kcals per day. Which you would think would not be possible, but medically supervised weight loss in this case, this case study, from 456 pounds to 180. That persisted for 5 years post-fast. You probably learn a lot about yourself and you develop some pretty strict habits over the course of a year of not eating. And few disruptive changes. Blood glucose is the top one. That's going to be really loud on the video because I just spoke right in the microphone. Progressive normalization, and even in the second half of the fast, no progressive decrease in blood glucose, highlighting to Leanne's point that there's an abundance of energy available to be able to make glucose from amino acids and fats. There's a whole host of other electrolyte changes and some other things, but the point is large decrease in weight. Oh, yeah. No fecal collections were made, but evacuation was infrequent. 37 to 48 days between stools.
Well,
I found that anecdote kind of funny as I read through it. It makes sense, though. There's no food in. Why would there be any waste out? Question, yeah.
wondering if these people are getting paid a lot, or do they just really want to lose weight?
I
Maybe this
honestly
person's
have
really
no idea.
Motivated, but even people in the last one.
I'm sure the last study would have had some financial compensation. I don't know. This is not a study that would have ever been that you would have recruited for.
Yeah.
And I think that it would have had to pass ethical approval, but it would have been after the fact. No one would say, oh yes, go and search out one person that's fat that you want to have not eat for a year. I'm sure, I don't know the specifics, but I'm sure this person probably contacted or was admitted to the unit, and maybe it was a personal goal of theirs to lose this much weight. But the Scots are also a little bit crazy, so I'm not sure.
So pretty much just saying that like this person had enough energy already on the body that they didn't need to eat for the full year?
Yes. Yeah. So I think, if I remember correctly, they had ample fluids and multivitamins available, but no food with any energetic cost. Complete fast minus multivitamins. The most extreme example, which I'm not going to go ahead and recommend to any of you, none of you need it. So the video that I want to watch, I'll put online, or I'll put a link online, but has to do with fecal transplants. So I'll skip the video for now because we only have 10 minutes. And I've affectionately named this slide transfusion, which is a mix of transplantation and something else. But there's some encouraging work here to suggest that implanting fecal bacteria or gut microbiota from healthy lean individuals into an obese individual can acutely improve insulin sensitivity. It doesn't necessarily fix obesity per se, but if diabetes is our focus, this can temporarily cure diabetes. Or no, let's say temporarily improve insulin sensitivity is a better way to put it. And this was a fairly recent study. This is only a year old, so this is still in its infancy, comparing individuals doing a self-transplant or self-infusion. And that's not to say they did it themselves. That's just to say it was a sham treatment where some of their fecal bacteria was extracted, mixed, emulsified, and then reinserted. They didn't know what they were getting, but this was the control group, the self-infusion group, versus those that received a donor transplant. So this is insulin sensitivity after receiving lean donor feces or their own feces, re-injected, hopefully not orally, on insulin sensitivity over 6 weeks. It is a funny topic, but it's pretty striking that these results, and you look over here on the right, statistical improvement in insulin sensitivity as measured by glucose uptake at 6 weeks. Half of the individuals responded positively to a lean donor transplant. No individuals responded to a self-infusion, which should not happen, right? There's no change to the status quo. No difference at baseline. You might, this is not significant, but you might argue if they're more insulin sensitive, maybe they're more responsive. No effect at baseline, but there were half of the individuals that responded positively. The downside is at 18 weeks follow-up, this response had reversed back to normal. At 18 weeks, no more effect. And that could be because there's no habitual change, no lifestyle change, or maybe a minimal lifestyle change over the course of the 18 weeks. So the donor bacteria can only do so much. Or it could be that the method wasn't exactly, didn't exactly take. Who knows that the bacteria proliferated and then replaced the microbiome of the obese individuals. Also, what's not shown here is that the half of the individuals that responded were the least diverse. So if you take a sample initially and you measure the diversity of bacteria, if you have few different kinds of bacteria, you responded really positively. If you didn't, if you had a wide range of different types of bacteria, you were in the non-responder group. So it's discouraging from the point of view of the study, but it's encouraging that you can almost identify responders and non-responders before even doing the maneuver. You can take a quick sample, analyze the genome, and say, oh, this would be a good candidate therapy for you. Yeah, question. Leanne.
Why not analyze the fecal matter from the lead donors and then just like give the obese participants probiotics?
Why not analyze the bacteria of the lean donors and just give probiotics? I think it's in that you can't directly grow the bacteria that you want just by administering probiotics. So probiotics are only food for the bacteria, and you can encourage some versus others, but bacteria are quite adaptable to use whatever energy is available. So the probiotics grow the bacterial colony or the total population, but I don't know that we have the technology to say this food will only grow this type of bacteria and not others. 2, you'd need to have the desirable bacteria there initially to be able to grow that population, right? If there's one type of bacteria that does a really good job of making you insulin sensitive, if you don't have that at the onset, it's not going to appear out of nowhere. I don't know anything other than what I have here. I don't know the specifics behind this kind of maneuver, but I don't think that we have the ability to just say, you bacteria grow, you don't. It's always a competitive environment that it's hard to manipulate from the outside through just the oral administration of prebiotics. That make sense? Sure. Okay, let's summarize quickly, and I'll post the link to that video online. It's pretty interesting and described in a very nice way. So we've seen in this section the spectrum of obesity and diabetes, the spectrum of obesity and chronic disease. And I think, my opinion, is that whether you move up or down on that spectrum is largely influenced by total energy balance. Positive energy balance moves you up on the scale, and then there are consequences of storing more energy. Negative energy balance moves you down on the scale, and there are beneficial consequences of inducing that deficit. I tried to link for you dysfunction related to obesity to diabetes through 2 main mechanisms, lipotoxicity and mitochondrial dysfunction, namely the excess production of free radicals or reactive oxygen species. And to cure or reverse progression of obesity and chronic disease, the solution, the logical solution, but that takes a lot of effort, is to create a persistent negative energy balance or energy deficit through the combination, ideally, of diet or exercise so that you aren't pushing one to the extreme. While we did look at some extreme examples today, and over the course of the measurement periods, there were no adverse effects. Who's to say if there are adverse effects afterwards? The combination of diet and lifestyle is certainly a more desirable or perhaps easy to adopt method of creating that energy balance. And then while there is promise for alternative therapies like the fecal transplant, at least at this point, they're temporary, they're transient. They work for responders, and they work temporarily. It's certainly worth investigating this method further to see if maybe there is a way that we can only feed the good bacteria, or maybe there is a way that the transplant can be facilitated, or the effects can persist longer. But right now, only temporary. Yeah, Peter.
I'm wondering if giving the participant antibiotics beforehand would increase the chance of the fecal transplant being successful.
Interesting idea. Okay, so try to flush the bacteria first, then introduce a new colony of bacteria that won't have competition to grow, and maybe that would help them stick around and maintain the insulin sensitivity benefits. Entirely possible. The hard part is that the bacterial cells in our bodies outnumber human cells, quote unquote. And the relationship we have with them is largely symbiotic, so I'd be concerned about such a significant disruption to the bacterial population. But I get where you're going with it, and I think there's promise there. I just don't know how I would do it yet. Yeah, Aaron.
I think they already use fecal transplant to treat things like C. difficile, which is caused by too many antibiotics or overuse of antibiotics.
Okay.
It kills off the healthy bacteria in your gut, and then C. difficile can grow and take over, and then they use the fecal transplant to reintroduce the
Oh,
healthy
fantastic.
bacteria.
Good. Some nice context then. Yeah, so it's not only limited to this scope, but also other issues as well. Great point. Thank you. Okay. Let's leave it there. I'll post the link to the video online. We're out of time anyways. Have a wonderful Thursday. And